Viruses show up everywhere on the MCAT, but do you actually understand how something that's not even alive can hijack your cells so effectively? In the next few minutes, we're breaking viruses down into simple, high-yield pieces: their structure, how they replicate, and the classic tricks they use to infect and spread. By the end, you'll be able to quickly recognize viral strategies and answer questions with confidence instead of guessing. Let's make viruses finally click. Viruses are acellular infectious agents composed of genetic material enclosed in a protein capsid, sometimes with a lipid envelope. Unlike bacteria or eukaryotic cells, viruses lack organelles, cytoplasm, and metabolic machinery, which means they cannot reproduce or generate energy independently and must hijack a host cell to survive. The core of every virus is its genome, which can be DNA or RNA, single-stranded or double-stranded. And this classification is extremely high yield for the MCAT because it determines how the virus replicates. Surrounding this genome is the capsid, made of repeating protein subunits called capsomeres, which protects the genetic material and helps with host cell recognition. Some viruses also have an envelope derived from host cell membranes, embedded with viral glycoproteins that function in attachment and entry. A key test tip is that enveloped viruses are generally more sensitive to environmental conditions, like drying and detergents, while non-enveloped viruses are more stable and harder to destroy. You may also see questions asking you to identify viral structure diagrams, where remembering that capsids can be icosahedral or helical helps. Connecting this to immunology, those surface proteins on the capsid or envelope are antigens recognized by the immune system. Overall, the minimalistic structure of viruses explains both their dependence on hosts and their efficiency in infection, which is why understanding structure is foundational for everything else in virology on the MCAT. Viruses infect host cells through specific attachment and entry mechanisms determined by interactions between viral proteins and host receptors. This specificity is called tropism, and it explains why certain viruses infect only particular cell types or species. The process begins with attachment, where viral surface proteins bind to complementary receptors on the host cell membrane, much like a lock and key. For example, HIV targets CD4 receptors on T helper cells, which is a classic MCAT example linking microbiology to immunology. After attachment, entry occurs either by membrane fusion, common in enveloped viruses, or by endocytosis, where the virus is engulfed into a vesicle. Non-enveloped viruses often rely on endocytosis and then escape the vesicle to release their genome. A high yield point is that blocking receptor binding can prevent infection, which is the basis for some antiviral therapies. Once inside, the virus must uncoat, releasing its genetic material into the host cell cytoplasm or nucleus, depending on the virus type. On the MCAT, you might be asked to predict which cells a virus can infect based on receptor expression, or to understand how mutations in viral proteins could expand or restrict host range. This concept also ties into evolution and natural selection, as viruses that can bind more effectively to host receptors have a replication advantage. Understanding attachment and entry gives you insight into both disease specificity and therapeutic strategies. The viral life cycle includes attachment, entry, uncoating, replication, assembly, and release, with variations depending on the type of virus. After entering the host cell and uncoating, the virus must replicate its genome and produce viral proteins using the host's machinery. DNA viruses typically enter the nucleus and use host DNA polymerase, while RNA viruses often replicate in the cytoplasm and may require their own RNA-dependent RNA polymerase, which is a key enzyme the MCAT loves to test. Retroviruses are especially important because they use reverse transcriptase to convert RNA into DNA, integrating into the host genome, which connects to molecular biology concepts like transcription and integration. After replication, newly synthesized viral components assemble into complete virions. The release step differs based on whether the virus is enveloped. Non-enveloped viruses usually cause cell lysis, killing the host cell, while enveloped viruses often bud off, acquiring their envelope from the host membrane. A common MCAT question might ask you to identify which stage is targeted by a drug or to predict the consequences of inhibiting a specific enzyme like reverse transcriptase. This life cycle framework is essential because it provides a step-by-step map of infection and highlights potential intervention points for antiviral treatments. Lytic and lysogenic cycles describe two distinct viral replication strategies, particularly in bacteriophages. In the lytic cycle, the virus immediately takes over the host cell's machinery to produce new virions, leading to cell lysis and release of progeny viruses. This process is rapid and results in the death of the host cell, which is a key feature often tested on the MCAT. In contrast, the lysogenic cycle involves integration of the viral genome into the host's DNA, forming what is called a prophage. The viral DNA is then replicated along with the host genome during normal cell division, allowing the virus to persist without killing the host. Environmental triggers, such as stress or UV radiation, can cause the prophage to exit the lysogenic cycle and enter the lytic cycle, leading to active viral production. A high-yield connection is that lysogenic viruses can contribute to horizontal gene transfer, including the spread of toxin genes in bacteria, which ties into bacterial virulence and genetics. On the MCAT, you may be asked to distinguish between these cycles or predict outcomes when conditions change. Remembering that lytic equals immediate destruction and lysogenic equals latent integration can help you quickly answer these questions. Viruses exhibit high mutation rates and genetic variability, especially RNA viruses, which contributes to immune evasion and challenges in vaccine development. RNA-dependent RNA polymerases lack proofreading ability, leading to frequent mutations during replication, which is why RNA viruses like influenza and HIV evolve so rapidly. This genetic variability allows viruses to alter their surface antigens through processes like antigenic drift, which involves small mutations, and antigenic shift, which involves major reassortment of genome segments, particularly in segmented viruses like influenza. These changes can render prior immunity less effective, explaining why flu vaccines must be updated regularly. On the MCAT, you may encounter questions asking why certain viruses are harder to vaccinate against, or how mutation rates influence viral evolution. This concept also connects to natural selection, as variants that escape immune detection or replicate more efficiently become more prevalent. Additionally, high mutation rates can lead to drug resistance, making antiviral treatment more difficult. Understanding viral variability helps explain patterns of outbreaks, reinfection, and the ongoing need for surveillance in public health. It ties together microbiology, immunology, and evolution into one highly testable concept.